Thanks so much for having me. So I'm going to talk to you today about the work that we've been doing trying to develop pipelines and assays and trying to discover mechanisms and pathways to block emerging RNA viruses. And I'm going to highlight some of the work that I'm doing in the AViD program. I'm a part of the CAMPP AViD, where I've been working on a program on flaviviruses. And I'm also part of the READDI AViD, where I've been supporting the programs on coronaviruses. And I'll mention a little bit of some of what we're doing in those AViDs as well. Okay, so obviously in this audience, it's not really that important, but I really like to remind people that emerging and re-emerging viruses are all over the place, and we're infected by viruses from many different animals all the time. And that's in part because of the world getting smaller. And a lot of that has to do with, in part, climate change, where we're interacting with animals at a higher frequency, in particular arthropod-borne viruses, which I'm also going to talk about today. And of course, we know about the recent pandemic with COVID-19, and many of the emerging and re-emerging viruses are RNA viruses. And so we've been really have a strong interest in developing models to study these emerging and re-emerging RNA viruses. Okay, so from my perspective, and that's obviously what I'm going to be talking about today, is thinking about how viruses emerge and re-emerge, and what are the parameters that are important to consider. And so we all know that in order for viruses to infect new hosts, they have to have all the replication machinery, including receptors, and this is really important and a really high barrier for many viruses to jump between species. Another important parameter, which in the AViD programs we're not necessarily thinking as much about, but I think are just as much of a hurdle, is thinking about the antiviral pathways that can be engaged. So many hosts are not permissive to infection because they have really active antiviral pathways, in particular interferon-induced pathways and interferon-stimulated genes. And so we're really interested in understanding what the barriers are, and then what the genes are that are important to overcome. And so how do we define these, and how do we inhibit them? And so obviously, from the context of the AViDs, we're really trying to inhibit viral infection using small molecules. And so there's really three classes that we've been working on, and I'm going to mention compounds from all three classes today. So we've been, and others have been, trying to identify direct-acting antivirals. These are the largest class of approved antivirals that block the virus itself, and we'll talk about some of the classes. The one that I'm going to talk the most about are nucleoside analogs that can block RNA-dependent RNA polymerases, for example. Then there are the host-directed antivirals that target human proteins that are essential for infection. While these have been excluded from the AViD programs, this is a really strong interest in my lab and many others, trying to understand whether or not we can use agents that block host factors as potential pan-antiviral agents. And I'm going to talk today a little bit about nucleoside biosynthesis inhibitors that target the host biosynthesis machinery. And then the third class that we've been interested in are these immunomodulators, and thinking about whether or not we can boost early innate immune pathways in order to block viral infection. And again, I'm going to talk a little bit about this class as well. Okay, so the way that we've been doing this at Penn is through developing high throughput assays, where we have robotics and liquid handling as part of the high throughput screening core here, where we have drug libraries and genetic libraries that we can transduce or treat cells with. And we've been doing these assays in 384-well plates using liquid handling and robotics. And we've been largely using wild-type viruses, where we can infect cells and then use automated microscopy to measure viral infection by staining for viral antigens. So we can stain for viral antigens, counter-stain for nuclei, and then quantify the percent infection. And within the same well, we have an idea about toxicity. And so we can also exclude compounds, for example, that have toxicity even in the primary assays. And then we take the candidates from these primary high-throughput screens and do validation studies, dose response, potency and efficacy studies, usually across panels of cell lines, to really find things that are broadly active. Then we move into primary cell models. And so like the respiratory models that we've been using are these air-liquid interface cultures that are really beautiful and can really recapitulate aspects of the respiratory tract, mechanism of action studies, looking across variants, for example, in SARS-CoV-2, combination studies to try to identify synergistic combinations, and then into preclinical animal models. So we've been really sort of pushing things through this pipeline, trying to identify as many, for example, compounds as possible that are active against these emerging and re-emerging RNA viruses. So the libraries that we've been screening initially were these repurposing libraries, which everybody has been doing, trying to find drugs that might be active. Many of these drugs are host-directed, but there are also nucleoside analogs, which I'll get back to. And we've been collaborating with many groups to get as many libraries as possible to screen. We also have a number of diversity libraries in-house in the high-throughput screening core, and I'm going to talk today mostly about this nucleoside library. Okay. And then in terms of the viruses that we've been screening, we have assays, high-throughput assays for all of these different viruses in-house. And again, as part of the AViDDs, we've been studying coronaviruses and flaviviruses. But we think it's really important to have as many models as possible to compare and contrast activity of these different molecules. Okay, so as all of you are aware, so I'm going to start by talking about SARS-CoV-2. And as all of you are aware, when the pandemic first hit, we all tried to do assays to try to find antivirals. And so, the virus grows in vero cells. And it was very quickly noticed that cathepsin inhibitors and hydroxychloroquine and all the endosomal inhibitors could block viral infection in the absence of toxicity. So in all my slides, toxicity is in green and percent infection is in blue. And so you can see there's activity. And the TMPRSS2 inhibitors really had no effect. And that's because in the non-respiratory cells, TMPRSS2 is not even expressed, okay? And so there was lots of confusion about what might be active in vivo, in part because of the models that were being used. And so we and others have developed this CALU3 model, which is a human respiratory model that expresses many of the cellular factors that are present on the respiratory tract. And we showed, and many others, that the cathepsin inhibitors, hydroxychloroquine, et cetera, were not active in these, whereas the TMPRSS2 inhibitors were active. And so this is just an example of why it's really important to look in the right model if you're going to look at active small molecules. And just to say, we've also shown that camostat, this TMPRSS2 inhibitor, is also active in human iPS-derived AT2 cells, as well as these air-liquid interface models. This is qPCR assay. And importantly, camostat and the TMPRSS2 inhibitors are active against all of the variants that have been emerging. So this is something that's really important for SARS-CoV-2 in general. And so we've done all of our SARS-CoV-2 screening then in CALU3 cells to find both direct-acting antivirals as well as host-directed antivirals. And this is an example of about 20,000 or 30,000 drugs that we screened of these repurposing libraries. And we found a number of inhibitors that were in the category of nucleoside analogs, as well as a lot of metabolic inhibitors. And so we were really interested in further pursuing the nucleoside analogs, in part because we rediscovered remdesivir and molnupiravir, as well as some other nucleoside analogs that are used in other conditions. And so we were happy that we found the drugs that were already known. But I just want to point out that if you look across cell types, again, even though this is a direct-acting antiviral, it has very different efficacy in different cell types. So it's really active in CALUs and A549s and much less so in viros. And so even direct-acting antivirals can have really different efficacies in different cell types. And that's because they're prodrugs and they have to be phosphorylated up. And the enzymes that do that can be expressed differentially in different cell types. Okay. So these nucleoside analogs are known to be misincorporated by the polymerase. And so that's how they function to block infection, at least remdesivir and molnupiravir. It's a little bit more complicated. But the other major class that we identified were metabolic inhibitors, many of which are involved in nucleoside biosynthesis. And so these are small molecules that end up depleting the endogenous nucleosides that are used by the polymerase to make viral RNA. And we were not the first people to discover this. And so it's really well established that RNA viruses in general need very high levels of nucleosides in order to replicate in cells. And so by depleting the nucleosides, then you can really attenuate viral replication, as shown here. And so we were interested in whether or not combinations of these drugs might show synergy in cell culture models. And so we do these 2x2 dose response studies where we look at the interaction between different classes of molecules. And so here, this is molnupiravir by Brequinar, a DHODH inhibitor. And you can see really striking synergy is measured by Bliss analysis when we combine these two different classes of inhibitors. And just to point out, we've tested many other classes in combinations. Here's just showing you nirmatrelvir and molnupiravir, but none of the other classes that we tested showed synergy in this kind of an assay. So we were really interested in further pursuing that. So then we did combinations of these drugs also in these air-liquid interface cultures, either from the nasal epithelium from a company called Epithelix, or bronchial epithelium from a company called Matec. And what we could see is really striking combination effects when we combined either molnupiravir with two different DHODH inhibitors in both models. This led us to try this in vivo, and we worked with Matt Freeman's lab to use this South African model where we could infect wild-type mice, and we gave them either molnupiravir or Brequinar or in combination. And we were really excited to see that the combinations had reduced viral loads compared to the single agents alone. And really strikingly, we had differences in the pathology in the lungs where the combination was really much more healthy than the singles, as an example shown here. And so we think that the combination of DHODH inhibitors with nucleoside analogs may be a way to synergize and boost the activity, helping to treat in the future. And so just to say that I think that this is not just something that we see in SARS. I want to give you an example with rubella virus. So this, we all have heard of rubella virus. Hopefully all of us have gotten vaccinated, and the vaccine is very efficacious, but there's still thousands of cases yearly, and there are no therapeutics. And actually, it's a live attenuated virus, so there's a subset of individuals that have disease after getting the virus. They're immunocompromised individuals. And so working with a clinician here at Penn, she requested that we screen to try to find inhibitors that might help her to treat her patients with the vaccine-derived infections. And so we screened a smaller library of 5,000 repurposing agents and validated about 20, and we validated them in A549s, which is a respiratory model, as well as in JEG3s, which is a placental model, because the real issue with wild-type rubella is neonatal infections. And so here's just two examples of candidates that we identified from the screen. We identified two nucleoside analogs that have been developed by other companies for HCV. We identified NM107, which is a pyrimidine analog, and AT527, which is a purine. You can see really good activity, complete block in titers. They work in all the cell types that we tested. And we're really excited to try to move forward with these. And so again, we did these combination studies where we did this pyrimidine analog with the pyrimidine inhibitor or a purine biosynthesis inhibitor, and vice versa with AT527 with either a pyrimidine and purine inhibitor. And we do see synergy, but interestingly, it's matched, which probably makes sense that the pyrimidine inhibitor works better when you deplete pyrimidines, and the purine analog works better when you deplete the purines. And so I think that this is a really generalizable concept in antiviral testing. And so as part of the READDI program in AViD, we've been trying to identify antivirals that are against pan-coronaviruses. focusing on nucleoside analogs, given the great success in identifying nucleoside analogs that can work against different viruses. And so we have this library of 1,200 nucleoside analogs that we screened against SARS-CoV-2, MERS, and 229E. And here you can see the data from the primary screen, where we screened SARS and CALUs, CAICOs, and A549s to try to find things that are active in diverse cell types, as well as screened against MERS and 229E. And so we're now characterizing the small number of nucleosides that are active across all these cell types and these different viruses to see if we can identify other candidates to move forward in the pipeline. And as part of the CAMPP AViD, we also have screened this library against dengue and other flaviviruses, and I'm just showing you some of the data here where we've screened dengue across cell types, so A549s, Hep G2s, IMR90s, and HY7.5s. The details here I'm not sure are that important, but again, you can see that these different nucleosides have very different activity in different cell types. And so again, we're really focusing on those that are active across cell types, in particular for the flaviviruses, because in vivo they can infect many different cell types in humans. And so again, we're trying to determine the mechanism of action and the PK and other things about these nucleosides to see if any of them are worth moving forward on. And so I just want to talk a little bit at the end here about our efforts trying to identify immunomodulators as a potential pan-antiviral strategy. And so, it was really clear early on in the SARS pandemic that there was really a delayed interferon response. And so you can see that, for example, in our experiments, where it takes a long time for the induction of interferons. But it is also clear that interferons can block infection. If you treat cells with type 1 or type 3 interferon, you can get reductions in infection. So while the induction is delayed, if you could get activation earlier, potentially that could block infection with SARS-CoV-2. And so we reasoned that maybe we could identify additional immune agonists that could block infection that were small molecules that might be beneficial over having to use interferons themselves, which are proteins and hard to scale and hard to deliver. And so we put together a library of 75 PAMPs, or immune agonists, against different pathways that activate immune signaling. And so we screened that in these CALU3 cells against SARS-CoV-2, and we identified a number of candidates that could block infection. And two of the candidates that we could really readily validate were cyclic dinucleotides. And cyclic dinucleotides are known to bind to STING to activate interferon signaling. And so we moved from the endogenous ligands to a small molecule called diabzi that was developed by GSK as a really potent STING agonist. And you can see here in these air-liquid interface cultures that we could see a really strong block in infection if we treat with diabzi or type 1 or type 3 interferons. This led us to move in vivo to ask if we could block infection. And we worked with Mike Diamond's lab to look at mouse infections treating with diabzi at different time points. This is just one example of some of the work that we did, where we could block pathogenesis in the K18 model, and we could really block viral replication in the lungs, suggesting that if you could give these immune agonists early, you might be able to really mitigate disease. And so what we think is that if we could come up with additional activators that could really activate immunity early, we could really block load. And this really might be very pan-antiviral, because most viruses are sensitive to the activation of interferons. And so we're trying to move this into different models to ask which viruses can be blocked in this way, and would there be benefit. And the last thing I just want to say is we did a screen. We've screened additional viruses with this PAMP library, and I want to just highlight one experiment we did with the Bunya virus. So Bunya viruses are also emerging viruses, and one that's really important is Rift Valley fever virus, which in Africa is really problematic, in part due to its high mortality of pregnant cows and sheep that leads to large numbers of abortions. And so in humans, it can cause a different febrile disease, but in about a few percentage of human infections, the virus is encephalitic and can infect the neurons. And so we were trying to develop models for Rift Valley fever virus, comparing neuronal infection to non-neuronal infection. And we made a really striking observation, which is if you treat non-neurons, and this example is U2S cells with interferon, you can really block infection. But in rat and other primary neurons, treatment with interferon has no effect on infection. So the virus is completely insensitive to interferon in neurons. And so we reasoned that maybe there would be other innate pathways that would block viral infection. And so we screened that same PAMP library that I told you about with SARS-CoV-2 in primary rat neurons against Rift Valley fever virus. And what we saw is that two ligands could block infection quite well, one of which is PAM3CSK4, and the STING agonist had no activity in this model. And so you could see here in the IF experiments that we could really block infection of these primary neurons with PAM3CSK4. We could also block another bunyavirus, lacrosse virus, but a similar ligand called PAM2CSK3 had no effect. And so we did a lot of experiments to figure out the mechanism, and we think it's actually blocking viral entry. But we worked with Amy Hartman's lab at Pitt to ask if this molecule could block Rift infection in an animal model. And so she infected mice with Rift Valley fever virus and also gave the animals this PAM3CSK4. And what I hope you can see here is we could very, very significantly protect the animals from death using PAM3CSK4. And strikingly, we also saw a really striking reduction in viral replication in the brain, suggesting that this molecule might be useful in blocking encephalitis. So with that, I just want to say we're trying to develop more assays with all these different emerging viruses across cell types and trying to develop primary models to have a better sense of which small molecules might be active in vivo during infection. And with that, I'd just like to thank the folks in my lab and the High Throughput Screening Corps and all of our collaborators from the AViDDs as well as other institutions who really helped us to move this program forward. Thanks.